Okay, so let's finish this up, and then I'm going to give you what I promised you. Do you remember what I promised you last class? Take-home project. So let's finish up this chapter, and then I'm going to give you your take-home project.
You told us to finish this chapter at home.
I know. Well, we're going to finish it in class. And you took your quiz, right?
Yep. Yes.
Did good?
I did 105. I
did terrible. I don't know how I did any good.
It was so hard. It was
Should have hung out with her.
the hardest. And I was down to the last seconds, and I'm like going, oh my God, there was some that I just won it. I just guessed at it. And I was like, how did I get 105?
Because you went with your gut.
Because I was like, it was so hard. It was the hardest one.
And you got 105.
I know. When I looked at it, I'm like, I was jumping around my kitchen.
Jumping around the kitchen. Okay, so these guys are probably jumping around your kitchen too. Who are they? Phage virus, yes? And what does a virus consist of? Yeah, a protein coat on the outside, and then some sort of nucleic acid, right? RNA, DNA. And they take over their host cell by doing what? Correct. And cause the host to start making more of them. So they have two reproductive cycles. One's called the lytic cycle. And this is where we're going to see many copies that are being produced. And in the case of a phage virus, we're talking about a bacterial host. Are there viruses that attack eukaryotic cells? Yeah, you probably are experiencing some of them right now. Some of you have the sniffles, feel a little crappy. It's probably a virus. And then the bacteria lyses, breaks apart, and that's why we call it the lytic. Then we have something called the lysogenic, and that's where the DNA actually inserts into the bacterial chromosome, and then the bacteria are going to reproduce normally. It's not going to kill them, but they're also going to make copies of what? Yeah, they're going to make copies of that phage virus's DNA with every cell division. So you're going to protect the integrity of the cell if you're in the lysogenic cycle. If you're in the lytic cycle, that's sort of the emergency. I have to reproduce lots and lots of me because I think that my host is not going to be able to do that for me. So in any given stage, we can be in the lytic cycle or the lysogenic cycle depending on the urgency of reproduction. And we talked a little bit about herpes virus and people with cold sores. Yes? So when do you usually get a cold sore? Yeah, something stresses the host. You're either sick or you're stressed out. Something stresses the host, and that causes the virus to kick it up as far as reproduction goes. So in this little diagram, we see a phage virus. This one's called phage lambda, affecting a bacteria, E. coli. And the genetic material in E. coli is not as highly organized as it is in a eukaryotic cell. So it's not too hard for that phage to get in there, cause the organism to make more of it, and release it into the environment. And we see that in your textbook.
So that one is going to the lysozyme stage?
Yeah.
Because it burst open?
It bursts the cell. The lytic stage, the lysogenic, is going to be a little bit easier. You're going to release virus. We'll see a little video in a second. Correct. So viruses that infect plants tend to stunt their growth, decrease their yield. Most of the known plant viruses have RNA rather than DNA as their genetic material. And one of the most commonly studied in genetics classes is something called the tobacco mosaic virus. Again, because it's very simple, very easy to see, highly reproducible, and we can study its genetics quite a bit. So, viral plant diseases have no cures. Viruses have no cures. Because can plants carry on an immune response and make antibodies against a foreign substance?
I don't know. They are eukaryotic cells, so why not?
Because they don't have the same systems you do. So they can't do what you can do. You can get better from, you can cure yourself of a virus by doing what? Yeah, those white blood cells are going to make antibodies, chemicals to neutralize the virus. The plant doesn't have the ability to do that. We can do it on the outside, but the plant can't.
If I walk up to a tree and whack it with something to take bark off, the tree continues to live, and eventually that spot heals.
Yeah. That's different. What's a virus do? Yeah, it's attacking the genetic material.
They make something that attacks the virus.
Because they don't have the...
Example of the virus, why can't they synthesize that?
We can, but the plant can't. Do you see where I'm at? So that we'd like to work on things to help plants cure themselves like we can cure ourselves. Correct. Or we can give them genetics to help fight off. You ever hear of genetically modified plants, genetically modified organisms?
You don't want to eat all that stuff.
Well, you do already, believe it or not.
No, I know.
So, no cure, best prevented by producing plants that can resist viral infection if we genetically modify them to be able to resist them. So viruses that infect animal cells are a common cause of most of the diseases we have. So most of the time when we get sick, it's not a bacteria, it's a virus. May have DNA, may have RNA, very varied when we talk about animal or eukaryotic viruses. Many animal viruses have an outer envelope made of a phospholipid membrane and little spikes of protein that project. Now, why do you think phospholipid membrane would hide it better? Right, because our membranes are made of what? Phospholipid bilayer.
I don't understand why proteins are all spiking out like that, though. Is that just to grab a hold of something?
No, that's just its cellular makeup. Well, it's yeah, I guess we could call it a kind of cell. Those protein spikes tend to be receptors for different proteins that are on the surface of cells. Yeah.
So that's what it's used in.
Yeah, or a door in. So, reproductive cycle of an enveloped RNA virus can be broken down into 7 different steps. So, this guy, it's phospholipids sort of fuse with the phospholipids of its host cell. So this is the host cell down here. Then what they do is they inject what? That genetic material along with some proteins into the cell. Those proteins disappear, uncoat, go away, whatever you choose to call it, and reveal the DNA or RNA. That RNA is then going to be synthesized with the help of enzymes by our systems. And those enzymes come from the virus themselves. That synthesis is then going to produce RNA, messenger RNA, a template for the virus just like we did transcription and translation of our own information, with the help of some of the enzymes inside the viral genome, we can do the same thing for the virus. So RNA strand is going to produce new viral genome, and then we make proteins based on message that was carried by the virus, the same way we do protein synthesis for ourselves. Make a messenger RNA, transcribe on the ribosomes, and create viral proteins. They'll then come together again and do what? Yeah, get out of the cell. So because they're coded, they are going to incorporate your phospholipid to help with the coating on the outside and leave. Genius, right? So, do flu vaccines protect the elderly? Yes.
Yes and no. No, yes, they do.
Yes, because I have to say that to pass this class, but no, because I know they really don't.
They don't. Now, how do you know? Based on your research?
Yeah, all my research
Okay.
is life.
Okay. So vaccination rates among the elderly rose from 15% in 1980 to 65% in 1996. So do they actually decrease the mortality rate among elderly people who receive them? What's the mortality rate? Death rate. Elderly people who were immunized would have fewer hospital stays and deaths during the winter after vaccination. So that's the hypothesis.
There's too many variables that could cause those people to live a little bit longer for that test to be conclusive.
Okay. Tens of thousands of people over the age of 65 were followed during the 10 flu seasons of the 1990s. And again, humans are tough because there's a lot of what? No, variables. But people who were vaccinated had a 27% less chance of being hospitalized during the next flu season and 48% less chance of dying. Those are pretty decent statistics. Yes, sir.
Isn't there like different types of flu though? Like
Correct.
obviously there's like H1N1 and like stuff like that.
Yep.
Like type 2 influenza, and is there? You get vaccinated for all those when you get
No.
the flu shot? Just one?
Nope. You were here. Were you here when I explained how it works?
Well, they take. I know that they like take virus and we can.
Well, that's how they make the actual immunization. But what they do is they look at statistically which are common in the area, or what they think are going to be common for the flu in the following season. So they guess, and that's a guess. It's an educated guess, but it's a guess.
So,
And they pick a couple, 2 or 3 different ones, and that's what you get in your flu shot every year.
So two
But
or
that's,
three. You get
yeah,
2 or 3 different viruses.
correct.
Okay, because I was gonna say.
So you don't,
I get the flu up
but
there,
here's
so
the deal. There's more than that floating around.
how many different types of flu viruses are there? You know?
They mutate all the time.
Probably billions of them.
But not at necessarily flu, but viruses that are going to make you sick like the flu. There's so many different types of viruses. And not all people who are very sick have the flu. I love when I get I have the flu. Well, you don't know you have the flu. You could have some other virus. It's not necessarily the flu. You just know you feel like crap, right? So it might not necessarily be the influenza virus. There's so many of them out there. Does that make sense? So here's some statistics. Deaths, hospitalizations. When do we see the biggest help? During winter months, when people tend to be compromised by other things as well. So the flu vaccine tends to be most beneficial to those patients during the winter months. Do we see a huge difference in the summer months?
Yeah, why do you suppose it spikes in the winter?
Why? Why do you think?
Because they close all doors.
Yeah, or the cold sometimes compromises people as well. Depends. You have to look at what we're talking about.
Maybe.
the different groups of people. Well, you know, I
Cold and heat, the change from hot to cold, I don't think has a lot to do with whether or not you catch a virus.
no, it doesn't have, but it has to do with compromising your body and allowing it to do its thing. That's why when you said to me the other day, every time I get the flu vaccine, I get the flu. No, you don't. But
With 3 different flu strains, you get the flu.
you don't get the flu. What happens though?
Well, you build antibodies.
You build antibodies against what they gave you, and while you're doing that, somebody might slip in the back door and get you from the backside,
Yeah, no, not.
right? Because you're compromising yourself by having that vaccine. But the consequences, seriously, after I took that emerging infectious disease course, I try never to miss my flu vaccine.
Does everyone get sick that gets the flu shot?
No,
I didn't get sick this year.
and they don't get sick from the shot, is what I'm trying to say.
It's just your body's reaction.
It could be something else that's making you sick. So, another very nasty virus that you've probably all heard about, devastating disease called AIDS, caused by this virus, is caused by something called the human immunodeficiency virus. Now, this virus particularly targets cells that are responsible for helping you get better during an immune response. So it's an RNA virus with some special little twists. It's what we call a retrovirus. RNA virus that reproduces by means of a DNA molecule. So it kind of works backwards. Retroviruses use the enzyme reverse transcriptase to catalyze reverse transcription, process of making DNA on an RNA template. So they go from RNA to making DNA, the reverse.
But the RNA isn't a double helix, it's a single.
Correct.
And then they use that reverse
Transcriptase.
taste to make it out.
Yep, so they'd work backwards.
But
And that's why they call them retroviruses.
Is it a double helix now that it's a DNA?
Well, that DNA is then going to turn around and make its RNA. So it creates the template so that we can make more of it. Backwards. Reverse.
It doesn't make a double helix, what you're saying.
Well, it creates the DNA,
So a double helix.
correct, which is now the template to make more of it.
Right. So
But it comes from RNA and works backwards.
yeah, but then it's going to reverse its direction where it falls.
Correct.
After it makes DNA to keep reproducing whatever it is.
Correct. So, reverse transcriptase, did that make sense? Does that
Yeah, no, it
make
makes
sense?
to me.
Okay. And this is what it kind of looks like. Again, it's an enveloped. It's got that nice little phospholipid. Behavior of HIV nucleic acid in an infected cell can be broken down into 6 steps.
Understanding the structure and life cycle of HIV, the AIDS virus, is critically important to dealing with the disease. HIV is covered by an envelope derived from a host cell membrane. Glycoproteins studying the envelope recognize and bind to receptor molecules on the host cell. A protein coat surrounds the viral genetic material, which consists of 2 molecules of single-stranded RNA. The enzyme reverse transcriptase enables the virus to make DNA from an RNA template, a trick for which this group of viruses is named, retroviruses. The RNA molecules of HIV enter a host cell when the virus fuses with the plasma membrane and the coat proteins are removed by enzymes. Reverse transcriptase catalyzes the synthesis of a DNA strand complementary to the viral RNA strand, and then a second DNA strand complementary to the first. The double-stranded DNA is incorporated as a provirus into the host cell's chromosomal DNA, where it may lie dormant for years. Occasionally, the provirus is transcribed into RNA. This RNA serves as both messenger RNA for the formation of HIV proteins and as genetic material for the next generation of viruses. Protein coats form around viral RNA and reverse transcriptase molecules. Viruses bud from the host cell, acquiring envelopes as they leave.
So these guys have the best of both worlds because they don't kill the cell. They take it over, and when they want, they cause it to make more of itself. Okay?
So,
The problem is,
when it's in your DNA, that's what AIDS is, but it becomes HIV when it starts that process of like making.
no, HIV is the name of the virus.
All right, so what's the difference?
AIDS is the disease it causes, acquired immune deficiency syndrome, when it's active. Because when it's active,
Takes over all.
it doesn't allow the cell to do what it needs to do. And one of the very important things that that cell does for a living is directs your immune response. So basically when something foreign comes into your body has a whole series of things it does to protect you from it. One of the things it does is very specifically makes chemicals called antibodies that neutralize those foreign substances. This cell, and it's called a CD4 positive T cell, is responsible for making sure that everybody knows exactly what it needs to do in an immune response. When this cell can't do its job, nobody else knows what to do.
So whatever viruses out there you get.
I don't know what to do. I don't know how to fight it. I don't know what to do. I call it the cruise director of the immune response.
Sterile bubble will stay alive.
Correct. Or something ridiculous can kill you. Something that we normally could fight off very easily if those cells were working. So HIV is the virus. Now, can you live for long periods of time with HIV? Yes. Can you live a long time with AIDS? Once these guys are active and no. So what the goal is, if you're HIV positive, is to do what? Control those guys so you don't get them active, so they don't start messing with your CD4 positive T cells. Yes. So when you take my anatomy class, we'll talk about an immune response. And you'll remember this conversation that we had today. So, viroids. There's two classes of pathogens that are actually smaller than viruses. One's called the viroid, and they just have a little tiny circle of genetic material, usually RNA, and they tend to infect plants. And another one that's even scarier than all those guys is called a prion. And a prion is basically a protein. And it, in the presence of normal proteins, can cause normal proteins to fold abnormally. Ever hear of mad cow disease? That's a prion disease. So, prions are responsible for neurodegenerative diseases. What's that mean? Messing up your nervous system. Mad cow disease in sheep and goats is called scrappy. Chronic wasting disease in deer and elk. And then in humans, Khrushchev-Jacob disease.
What does it do to humans?
Same thing. Degrades your central nervous system.
So what does it do?
It's going to kill you eventually. Central nervous system is basically your controls.
Yeah, I know that, but like, I would say like what the symptoms are, you know?
Well, anything that degrades nervous system, so it depends on where it starts, it depends on its origin. You'll start to see problems with you being able to function properly. Muscle control, speech, thought. And again, depending on where it starts and moves, would change the symptoms. So don't eat the central nervous system of things. Where these strange yes. And that's how you spread it from one to the next. So if I take these abnormal proteins and I feed them to another cow, and how do we feed our cows these days in feedlots? Do we feed them what they should eat? What should cows eat? Grass. Cows eat grass. But what do we feed them? All kinds of crap. To do what? Fatten them up fast so we can get them to market and make some money, right? Because it takes a long time for a cow to get fat on grass, right? So we feed them things that we shouldn't be feeding them, like ground up other cows.
Didn't they do that when they had the mad cow disease? Didn't they grind them up and then feed them to the new cows? Yeah, I thought I heard that.
Before they realized what was causing it. So the cow would fall sick. They're like, okay, I'm going to take this cow and grind it up and feed it to my other cows and make them fat quick. Yeah. And then that Khrushchev-Jacob disease was actually seen in tribes who had rituals where they would eat what?
Brains.
The brains and spinal cords of their dead.
We don't eat that. We don't have that.
Hopefully you don't eat humans.
Well, they're dead?
Well, it's in
Oh, that's messed up.
we try not to get it in there, but
What happens in like 5 falls into a grinding machine. Nobody notices for a couple of weeks.
so the evolutionary connection, emerging viruses. Viruses can reproduce at a very fast rate, and they can mutate at a very fast rate. That's why they're such a pain in the keister. Viruses have suddenly come to the attention of science because every once in a while one of them is very virulent and makes us really sick. H1N1 and the avian flu happen to be on the forefront, at least right now anyway.
So why was H1N1 such a big scare?
Well, because it was very virulent, a lot of people were getting very sick from it.
I got very sick. My grandfather caught it when it was outbreaking like that.
Yeah. But that's why, because it just made you that much more sick than the normal whatever was around. And that's what virulence is. So avian flu infected birds and then jumped from birds to people, because typically viruses like to hang out in certain type of cells. But once they start to mutate, they can jump from one species to another. And that's what happens with our flu virus every year. Because the flu virus comes from mutated virus that tends to hang out in birds and pigs. And then it jumps to a few humans. And then those humans do what? Will spread it to other people.
Do you have to eat the bird?
No?
How do you get it?
Well, you can be in contact with it. Typically eating it isn't how you get it. You live in close proximity to. And we typically don't live in unless we're farming, we typically don't live in close proximity to birds and pigs. But where do people do that?
Just touching it like this slime.
And breathing. A lot of them are respiratory viruses.
People who work in farms too, they don't get bothered by
Or if you live in Asia, or where do you put your ducks and your pigs? Yeah, they're usually living right quite close to you. They don't have the luxury of the farm and the barn. Yeah.
That's the barn.
Yeah, they're walking in and out.
They're not always washing their hands.
No food.
Oh, I gotta get that. To stop out on my tooth right after I get done messing with a pig or.
You get it. That's the way you get it. So when it jumps to people, then we go cough all over everybody else and spread it to the rest of the people. 18 people. That's all it took in Hong Kong in 1997, and then spread to Europe, Africa, infected over 400 people and killed more than 240 of them. That's a big,
Percentage.
yeah. See that number? That's a virulent virus. It tends to make you more sick than others. Do you see what I mean? So that's when that comes up on the radar, especially when we see a lot of deaths from such a small amount of infection. That small group very quick. No.
I don't remember a lot of people dying from H1N1.
There was a higher mortality rate, and it was on the radar because of the avian flu.
Like cause if you get like pneumonia and they're among those people.
Yeah. Oh yeah. Over 100 billion birds were either died from the disease or they were killed to do what? Prevent the spread. So, avian flu can mutate to form something that can spread very easily to people, and then the potential for major outbreak in humans is significant. So, how do we get new viruses? They arise from mutations. Mutations of existing viruses. Or the spread of existing viruses to a new type of host. So sometimes certain viruses might not come in contact with this new host, and we think it can't jump, but then when it comes in contact with this new host, it can. So just exposing it to the new host, allowing it to jump from one species to another, can also have a devastating effect. Do we understand how viruses work? Do we understand their genetic material? Are we ready for our take-home quiz? Or take-home test?